Today, what I'm going to do is talk to you a little bit about one of my projects that focuses in on water. My lab does a lot of different things. And so, talking to Zach, what I'm going to do first is, because many of you, even though I've been here for a long time, I haven't given a talk on campus in probably a decade. I'm going to talk to you a little bit about who I am as a scientist, how I approach my work, and then I'm going to tell you a little story on how we got this cellulosic biofuels project going and where we've taken it to date, and then talk a little bit about where we're taking this project in the future. So I would consider myself an interdisciplinary scientist. I was classically trained in graduate school as a plant physiological ecologist, but over my career, my science has really evolved, and I focus a lot on ecosystems ecology and understanding biogeochemistry and the linking of biogeochemical cycles. And what you'll see from this project, I do a lot of micrometeorological and atmospheric science measurements to try to understand how whole ecosystems take in or give off various types of greenhouse gases. And one of those, of course, is water. So my lab is the Global Change Ecology Lab. And we really look at the coupling of biogeochemical cycles in both terrestrial and aquatic landscapes. And I'm really interested in the anthropogenic pressures that are being put on ecosystems that alter their structure and function, which leads to changes in this land atmosphere or aquatic atmosphere exchange. And in addition to this, we are studying natural climate change solutions in our most recent project. And currently, my research is focused in on basically 4 areas. We really focus in on forests of the southeastern United States, our biofuels studies. I also work in Alaska in the Arctic tundra. And I have an extensive long-term project that's been running 16 years in Everglades National Park, looking at how changes in freshwater inputs are affecting these different biogeochemical cycles. And so, as an ecologist, most scientists in my field either take top-down or bottom-up approaches to their science. I really consider my approach a middle out. I focus in on ecosystems at the center of this scale, and most of my measurements are at this ecosystem level, but then we try to deduce what components of that ecosystem are contributing to our ecosystem responses. So I might be studying trees or leaves or various quadrats within the study. And then because I'm working across a variety of different ecosystems across the South or even across the globe, we try to compile all this data and make inferences about landscapes, regions. Even I've been working recently on continental scale greenhouse gas emissions. And a few years ago, I worked on a large-scale study looking at productivity and the emission of CO2 for the whole northern hemisphere. So we work at a lot of different and take a lot of different approaches to our science in my lab. And so today I'm going to talk to you about the cellulosic biofuels project that we've been conducting since about 2014 at Savannah River National Lab. And I'm going to talk to you a little bit first about how did this project come to be, right? So really the foundations for this project come from the fact that the human population has been exploding since the onset of the Industrial Revolution. And with those increases in human needs and activities, there has been an increased demand for energy products and energy production. And although per capita fossil fuel use has decreased because of this rapid increase in population, our use of these fossil fuels continues to increase. And that's having an alarming effect on ecosystems and the planet as a whole. And we know this is occurring in many areas and at different rates. We're seeing that these rising concentrations of CO2 and methane are causing increases in temperature. And we know that our global temperatures have increased. Interesting too, After, in the mid-2000s, methane being kind of stable, it rapidly started to increase. And even with the pandemic over the last couple of years, we've had the 2 highest out of the last 3 years methane emissions in the world. And this is also having huge implications in our atmospheric chemistry and the climate on the Earth. So we are seeing these global temperatures rising. We're seeing an increase in extreme storm events. So heavier precipitation, greater droughts between these precipitation events. These are all affecting the water cycle in different ways. We're seeing with this warmer temperature, evaporative demands are changing on the planet. And the Earth's atmosphere can actually, as it warms, hold more moisture in the air, and this is affecting the global water cycles. And so, in addition to this, one of the interests we're looking at is that these warming events are causing changes in plant and ecosystem water use efficiency. So how much water is being given off per molecule of CO2 that is being taken in. And there's a lot of debate on if this is a sustainable change or if these are temporary changes when it comes to plant activity. And so based off of these changes, the federal government started to act, and in 2005, they produced their first major policies on creating biofuels. And they gave people grants, economic incentives, and loans to create biofuels and develop research. And they made this mandate of incorporating 7.5 billion gallons of blended gasoline into our economy by the year 2012. The problem with that was, in these early stages, they were really focusing in on edible biomass, these first generation biofuels. And even though there's 4 that were proposed to be used, we primarily as a country focused our energies on using corn as a biofuel and turning that into ethanol. That raised a lot of problems. But people were still pushing for these biofuels. They were saying, you know, biofuels can reduce our need for foreign oils. They can reduce our greenhouse gas emissions. They don't require as much energy to produce these. And so this is a win situation for us. But when we first started this out, our current average reduction in greenhouse gases was only 19%. So it wasn't very efficient. And we weren't taking into account all of the factors that were influencing our reduction in carbon balance. Since we were using edible crops, we created another problem. That problem was the debate on, are we trading food for energy sources, right? And so in addition to that, we were taking these viable agricultural lands and we were turning them into an energy crop. And that was causing land competition across the globe. I worked in South Georgia, and all of a sudden when these acts started popping up, people were putting in corn everywhere and using pivot irrigation to water them, which was another influence on the water cycle. Just creating these farms, you're using more greenhouse gases to run the machinery, so the balance or mitigating that carbon dynamics wasn't there. And then you're adding to grow these things, fertilizers, pesticides. This can have downstream effects with eutrophication of streams and so forth. And here's an interesting one. For every billion gallons of ethanol we've created from corn, we increase the price of corn 2 to 3%. So that initial stage of 7.5 billion gallons, we increase the price of corn between 14 and 23%, somewhere around there. And that has huge implications on food costs for everyone because of everything that corn is made with the high fructose corn syrups. So the government soon realized that may not be the way to go. And they quickly developed a new Energy Independence Security Act in 2017. And this really mandated us to push our research forward in a way to create new and alternative biofuels that weren't focused in on food resources, right? And so they, though, increased their demands and said, you know, we want 36 billion gallons of biofuels produced in the United States by this year. And then 21 billion gallons of that should come from cellulosic biofuels and advanced biofuels derived from feedstocks, which is part of the foundation of our research that we created. And we then started focusing in on these second generations of biofuels. And in these second generation in the United States, three main crops kind of came to the forefront. Loblolly pine up here, which is the focus of my research. There's switchgrasses in the center and poplars. And some of the arguments that scientists and the government were making was that these new bioenergy resources were grown on marginal lands not suitable for crops. And therefore, we could eliminate or at least reduce this food for fuel concern. But there were still questions. Are these really viable crops? And are we creating new problems by growing these different plants in such high densities? At the same time, the Department of Energy had created Beto, and Beto put out all these calls for new bioenergy technology. And at the same time, the Department of Energy went in on a new cellulose biofuels feedstock energy plant at Savannah River National Lab. Savannah River National Lab is 320 square miles of forest. So there's a resource right there that they were thinking they could take to that energy production plant and run the whole national lab on. And this was where our research funding originally came from. And since we were in the South, we focused our research on understanding loblolly pine. And the reason we did that was loblolly pine is the dominant timber product in the southeastern US. Currently, there's over 30 million acres of loblolly plantations distributed. Historically, we know that they have high productivity. They sequester somewhere between 6 and 10 tons of carbon per year per hectare. And originally, in the last 20 years, the rotation age was somewhere between 20 and 24 years, depending on what you were using those plantations for. Was it pulp or was it saw timber? But to get this to be a viable energy crop, modelers predicted that we had to reduce the rotation age by 50% and get it down somewhere between 10 and 12 years. So this means we had to rapidly increase growth of these trees. And due to the silvicultural history of using loblolly, many companies started to introduce relatively quickly new sibs of trees, new silviculturally selected for genetic stocks. And so we were fortunate enough to get one of those genetic stocks and plant it. But this, again, would only increase the growth to a certain extent. To get it to this point, we have to increase our intensity of management. So intensive fertilization, intensive herbicide, much more than a conventional pine plantation, which, as I said before, can lead to a substantial number of environmental effects. Nutrient pollution, eutrophication, herbicide affecting things. And then there's this question of, are we trading water for carbon? So Rob Jackson is a colleague of mine who is at Duke University, and he did in 2005 a bunch of simulation models of if we increased moderately, not even to the rate that we're expecting these loblolly plantations to go, what that would do to water resources in the South. And he showed that under certain dry conditions, that an increase in loblolly plantings would cause many streams in the Southeast to reduce their discharge or even go dry. So then that has implications in food web dynamics, the protection of endangered aquatic species. So this really set the stage on, is this a viable crop? Are we going to do more damage than good? And that's where my research comes in. So in 2023, after we got this large grant, a huge group of us, including the Forest Service, 5 universities, and Department of Energy scientists, put in this watershed at Savannah River National Lab, which is a 120-hectare watershed, a short rotation woody crop of loblolly with these improved trees. And this is the general water cycle, and what I'm going to focus in on today is understanding these components, the transpiration, the evaporation, soil moisture, and how those parts of the water cycle are changed with these genetically modified trees. And so this comes out of a paper of ours. We were a large, intense group, right? We wanted to evaluate the environmental effects of this crop. And so we looked at state BMPs. We made the decision to plant this woody crop. We followed the site through site preparation, all the herbicides, fertilizers, and we took on all of these different measurements all the way down. The only one we really didn't focus in on was biodiversity, and we wanted to see, is this crop environmentally sustainable to reach a goal of making this a biofuels feedstock? And so my group, cutting out our narrow window, looked at determining and evaluating what were the drivers influencing ET. And this is a whole ecosystem measurement. So we also wanted to decompose this, right? That's how I like to do my research. And so we looked at the different components of ET2 and incorporated a variety of additional measurements. We predicted that as the canopy increased, the components of evapotranspiration would change, and that soil evaporation would be less of a component of the ET, and that this rapidly growing canopy would have increased transpiration. And we also said that if these trees are really going to grow as quickly as we're projecting them to grow, it's most likely that this stand will be a much larger user of water than these conventional pine plantations that are distributed across the southeastern United States. And so this just gives you an idea of our approach. So we have the eddy covariance up there that measures the whole ecosystem. We have sap flow sensors that'll measure transpiration, and I'll explain those to you in a second. We have through-fall collectors, right? As the canopy develops, those leaves will just catch rainfall, and it'll never become available to the plants, and it'll be evaporated right back off. So we need to know how much of that rain is never reaching the ground. And then, of course, box lysimeter to see how much evaporation is coming off bare soils. And so, real quickly, these ecosystem measurements, eddy covariance, if you're not familiar with, wind flows through the atmosphere laminarly. Just like in a stream, when it touches something rough, i.e. vegetation, it becomes turbulent and makes these eddies of air. These eddies can be really small or very large in size. And within those eddies, there are specific concentrations of greenhouse gases. As those eddies dip into the forest, if plants are photosynthesizing, they'll draw the CO2 out of those eddies and replace it with water. And so we measure the wind at really high rates, 20 hertz, and the concentrations of greenhouse gases in those eddies. And we can basically then tell with a mass balance and a bunch of mathematics how much photosynthesis or respiration or evapotranspiration is occurring from a forest. And so we can tell the whole system, but then I wanted to understand what is contributing, how are those components changing as the system rapidly develops. So to measure soil evaporation, we use these lysimeters that we distributed throughout the forest. These were co-located with the rain gauge that was on the forest floor, and then one's above the canopy. And you could tell then, based off changes in the volumetric water content within those lysimeters, how much evaporation was coming off of them. And this is just a picture of one from our site. We also use heat dissipation sensors, or Grenier sensors. So you can go out and buy these sensors, but they're about $400 each, or you can make them for about $2 each in your lab. And so the idea here is you take bovine needles, you stick a microfine thermocouple in them, you drill two holes about 10 centimeters apart in the tree, you put a thermocouple in your top probe, and a heating wire. And you insert those into the xylem of the tree, right? The conduits for water movement. And you heat that top one. And as water moves, it will dissipate heat away from the bottom probe. At night, when the tree shuts down, you will have the greatest dissimilarity. And then you go in and you stain your active xylem, and you can get the xylem area of a tree, and then you can basically scale up and determine what a whole tree's water use is. And then we'll distribute these across the forest, and we can then model whole canopy transpiration from those. So the next step in separating these. And then the last thing we did was we had through-fall. We had above canopy rain gauges, and then we had these catches where we know the area that we are catching precipitation coming through the forest. We capture that and we record it through data loggers. And then we can tell approximately how much water the canopy is catching and re-evaporating directly off of the leaves or needles. But we also needed to do some ground-based measurements, right? How rapidly is that canopy developing? How much leaf area is up there? So we did hemispherical photos over the through-fall traps. So this is the development of the same spot, and in a little over a year, how much leaf area has accrued there. We can determine through some software gap fraction, so how many openings are in that picture of clear sky. We also did canopy height, which we can then use some atmospheric techniques to look at how decoupled the soils are from the atmosphere, and then some basic forestry just to see how much wood volume. And we ran this study for one of my students for 31 months, from April 2016 through October 2018, so years 3, 4, and 5 following planting. So still really young forest. So what do we see here? We saw first off on the top that these trees were growing really rapidly. So from 2016 to 2017, these trees put on over 5 centimeters of DBH. They put on over 2 meters of height growth, pine trees, in 1 year. And a year following that, we actually saw on some trees as much as 4 meters of growth in a year from a pine. And then these are some of the components which I'll talk about in more detail in the next slides. Does anybody have any questions? All right. And this is just to show you the rapid change in leaf area. So we went from a leaf area of under 1 meter, about a half a meter per meter of squared ground, all the way up to, in our most productive or highest leaf area plots, of 4.2. That's a 4-fold increase in 3 years in the leaf area of the system. And then we see that our precipitation followed a natural range, greater precipitation in the summer months or the growing season, less in the non-growing, except for that one tropical storm that hit our sites that one November. So what does this mean for water movement between this system and the atmosphere? These are just growing season averages, so 6-month averages, or sums of precipitation. This is our eddy covariance. And those are the components of the evapotranspiration. So we have soil evaporation, transpiration, canopy interception. What's that canopy? And what you see is, of course, a decline in the non-growing season when things are cooler. And although we had these intensive management of the system with lots of herbicide and that, stuff still recruited back into the site. So this wasn't just pure pines, which will tell part of this story. The big thing to note here is that in this third year of the study, we see a substantial decline in soil evaporation, an increase in transpiration when we really got that leaf area going, and a slight increase in canopy interception. And this just shows this on a more detailed basis here, right? This strangeness and canopy interception that was so much was during that tropical storm. Basically, the canopy was so saturated, the water just flowed through. And as you can see here, soil evaporation declined substantially in that last year. This also showed from our site and explains some of the unexplained portion of ET. When I showed you guys those components, right, on the last slide, eddy covariance was pretty closely linked to precipitation, but when we started adding up those different components, there's a portion somewhere between 13% of the ET that we can account for up to 33%. And so this just shows, when it's positive, that eddy covariance was indicating a much higher evapotranspiration rate than the components. And so what you see here is that during the summer months or the growing season, that there was a much larger dissimilarity in our measurements between the components and the eddy covariance versus the winter months, which are in yellow. So I mentioned, right, that other things grew in there, and that's going to be a big part of the story. A lot of these understory grasses and some deciduous seed bank was still there. In the winter months, those all go fallow, right, or senesce for the winter, and only the pine trees are active, and that allows us to have more similar measurements between our components in that. We weren't focusing in on the deciduous shrubs or the grasses in the understory. So one of the things we wanted to understand here was, well, what environmental variables, right, that first hypothesis, were important? We took 25 or 30 different environmental variables, and we ran to first determine how we could reduce the number of variables we would look at in this system by running a principal components analysis. And what we saw was that a lot of our variables tracked one another, which we would expect, right? So our different soil temperatures at depth tracked each other, which tracked air temperature, which tracked soil heat flux, our G. So we could condense those down and use only one specific variable as we went on to understand what were the key drivers of this. We also saw that there were different responses between the non-growing season and the growing season responses of this system. A lot of this, again, could be attributed to certain species senescing in the winter months. So we did a partial regression slope study on this. In the second column is our eddy covariance, so our bulk fluxes, and then our different components, our transpiration, our interception, and then our unknown portion, and our soils. And so some of the big take-homes here was that we see that vapor pressure deficit and growing season have a negative effect. So as the air got drier, evapotranspiration went down. And we know this from pines that at about 2.3 VPD, we have a stomatal limitation and the pines shut down. The air is just too dry for them to transpire. We also see that net radiation, right? The total amount of radiation coming in plays a role in heating the system. For the different components, VPD had a smaller effect on just the general ecosystem, but again, a negative effect, stomatal limitations occurring. And those were some of the big take-homes there. Some of those are very minimal effects. So we started to understand temperature and radiation inputs are two of the big factors. One of the other key events that we were concerned about, right, was how are our systems responding compared to these conventional plantations? And so we went out and we searched the literature, and we found every single pine plantation study. We even threw in some slash pine, which is dominant in the timber production in central Florida. And we started to look at differences. That won't show up. So ours is the top. And even our 5-year-old stand had a 3.1 leaf area on average. We used, when you go out here and look at evapotranspiration versus precipitation, we're using about, over the course of this study, about 82% of the precipitation coming in is being evaporated and transpired back off. In many cases, this is very similar to much older stands that are between 8 and 16 years old, like Soon et al. Showed in this lob lolly plantation in North Carolina. We also see that our plantation at 4 years old has about double the conventional plantation's leaf area as the other Soon site, and that we have much higher transpiration and evapotranspiration rates than a site that is of similar age to ours. So what we're seeing here is that this genetic improvement has really enhanced the early development and the rapid development of our stand compared to these older genetic crops of trees. And this is playing a huge role in how this ecosystem is responding. So that is the scientific story that we know to date. And so when we're thinking about this system, some of the things that we've discovered so far is that during early stand development, our soil evaporation rates dominated for the first 2 years of the study. They contributed anywhere from 23 to 37% of the overall evapotranspiration for the system. As leaf area increased, we saw that soil evaporation decline readily, and the contribution of transpiration with the increased LAI started to dominate the system, and interception increased too. We also saw that there was greater uncertainty between our methods when we were in the growing season. And as I said, we attribute a lot of that to the fact that we saw senescence of all these deciduous plants. The other thing that we can't account for that is really important to note is that there are uncertainties with every single measurement strategy. And we're measuring things at different scales. We're measuring 30 by 60 centimeter piece of soil. Our eddy covariance is a dynamic measurement that moves with the wind, and we might be measuring as much as a half a kilometer square of forest. Our sap flow sensors, we are measuring maybe 25 or 30 trees within the vicinity of the flux tower. how much variation is there occurring, even though we had different size classifications? This is something we're not sure of. But like I said, the difference between the eddy covariance and our component analyses were somewhere between 13% difference in the winter and as much as 28 to 30% in the summer. We saw that our evapotranspiration rates, even on this very young stand, were more similar to stands somewhere between 8 and 12 years old. The other interesting thing, if we just look at a rotation, we believe that we can reach our goal of cutting these trees at 12 years. And at a single rotation, our stand has slightly better water use efficiency. And one stand, these new biofuel trees will actually use less water, but we're only growing them for half the time. And when we scale this up over 100 years, right, that conventional piece of pine plantation that would have 4 rotations versus our biofuels land would have 8 rotations. And when you think about that, we're actually using substantially more water, and we're actually, on that 100-year time scale, most likely trading water for carbon. And that's the big concern there, right? And how this will affect our regional water resources if these biofuel plantations are expanded to larger areas across the globe. So that's where we are now. Where are we going with this research over the next couple years? One of the things we're really interested in is trying to understand those multiple rotations, right? They're improving the trees constantly. Will our next rotation have even higher quality trees that grow even faster? By doing that, will we be robbing the system long-term in the water yields from the soil to even a greater extent? We're also trying to look at, which no one has ever done, usually as we go to the period when they're going to cut the forest, everybody takes their equipment out for fear it'll be damaged. So we don't really understand how the system is responding during a large prescription treatment. So my goal is to run all our equipment while the John Deeres are in there cutting down, while they re-till the land and replant, to understand those windows that no one understands before and see how that contributes. And then I'm taking all of my component data and my eddy covariance, and I'm working with a variety of different hydrologists at the University of Georgia and Oregon State to parameterize the SWOT model and understand how variations in climate, extreme events, and just different management regimes will affect water movement, water yield, water quality at these sites over multiple rotations. The other thing we don't know that takes us a little bit away from the water story, we still don't really know, is this a viable energy crop from a carbon point of view? So I'm looking for a student who will potentially work on doing a life cycle analysis of the carbon. So it takes so much to produce fertilizer, right, from a greenhouse gas point of view. It takes so much to fly that helicopter to spray this crop, to remove it. And where are you trucking it from too, right? To move those logs off of the site, you're burning fossil fuel. And until we know all of those transportation costs from a greenhouse gas perspective, we won't really truly understand if this is a viable energy source for the system. And at that point, I'm going to wrap up. This was a huge undertaking. And I've talked about in our overall large project that this being associated with 5 universities and 2 government agencies, but from just this portion of the study, my lab had several people contributing, coming out to do field surveys, helping maintain equipment. Dr. Stodhammer's lab, her students were involved. My colleague Doug Aubrey's technician was involved in running a bunch of this. And then a variety of U.S. Forest Service individuals who helped us on site maintain, implement the prescriptions, paid for the prescriptions over the course of this time. And we had with this a variety of different funding sources. The University of Alabama was very instrumental by giving me some teaching assistantships, the Forest Service, Beto, and a variety of other cooperative agreements between the Department of Energy, our universities, and the Forest Service. And at that point, I'd be happy to answer any questions you have.